Hi, my name is Annie, and today I'm here to talk to you about my project titled Hints of Novel Immune Signaling Pathways. Eukaryotic serums are capable of NAD degradation. Now, that's a lot of words. And to be honest, when I first joined the Morehouse lab, I didn't know what any of them meant. In fact, I was freshly 18, and not only was I struggling to adjust to lab work, I was still figuring out how to be an adult. I wore mismatched socks every day, always forgot to pack lunch, and once had to bail on an experiment because I threw up due to food poisoning. At that point, I had no idea how small was that little bacterium ransacking my bowels. Bacteria are constantly under threat of infection from viral bacteriophages and have to evolve a diverse arsenal of defense systems to protect themselves. One of these defense systems is called the Thorax defense system, where, upon pathogen detection, a bacterial protein called Thisei will cleave the cellular metabolite NAD using a serum domain and execute immune response. The human innate immune protein Sirims also contains a serum domain and is able to cleave NAD. It is hypothesized that Sirims is evolutionarily derived from an ancient prokaryotic serum domain, inherited from the first endosomatic event that gave rise to the first eukaryotic cell, or soon thereafter via horizontal gene transfer. If that's true, we would expect to find NAD-cleaving serum enzymes in diverse eukaryotic organisms. To investigate this possibility, our collaborators employed a homology-based search to find other possible eukaryotic serums. Their work resulted in 9 possible eukaryotic proteins where we need to test for NADase activity. To do so, I purified these proteins and incubated them and tested with NAD. To observe for NAD cleavage, I separated the reaction products using high-performance liquid chromatography. Out of the 9 possible serums we selected, we identified 2 serums that were able to cleave NAD, one from zebrafish and one from plankton. We observed that these serums are able to split NAD into its 2 constituent molecules, ADP ribose and nicotinamide. But Annie, you might say, why should we care? Well, first of all, NAD cleavage has been implicated in other immune signaling pathways, such as with the human tryptophan-2 calcium ion channel or in plant immunity, suggesting that these new serums may be involved in previously uncharacterized signaling pathways. Secondly, zebrafish and plankton are from distinct sections of the eukaryotic domain, yet they both possess similar NADase activity. This strongly points to the presence of a serum in an early eukaryotic common ancestor, rather than just being a result of multiple de novo developments. And most importantly, just as immunity is ever evolving, I too am always growing. As an undergrad in the Morehouse lab, I have learned and experienced more than anything I could have ever imagined. Indeed, I sashay every day into work with matching socks, only occasionally heat up my packed lunches in metal bowls, and have ceased to DoorDash suspicious takeout before experiments. So to the Morehouse lab and our collaborators for helping me grow, thank you.